So, our next amazing speaker is Dr. Dept from Department of Plant, Soil, Microbial, Ecology. He's going to talk about cultural and biological control methods of critical disease in Michigan vineyard. Doctor, please. Yeah.
Thanks, Ismail, and thanks to everybody, P45, the Wine Collaborative, everyone else for inviting me. So it's a great opportunity to talk. Second. Okay. So I was trying to be a little bit funny with this talk, but we'll see how it goes. So thanks for attending, and I kind of miss not actually being up in Traverse City during this time period. So try to make it as interesting as I can. The title of my talk today is looking at cultural and biological control methods of critical diseases in Michigan vineyards. And I pulled this off of a show that I watch at Star Trek. But basically, even in the future, this is supposed to be a futuristic representation of a vineyard, probably in France, but it looks like it's actually in California. But it seems that fungicides still may be needed in vineyards in the future. So anyway, my name's Tim Miles, and yeah, I'm a plant pathologist. So before I really get into the talk, and this talk's really not very research oriented. It's a little bit, I've got some trials I'm going to show at the end, but it's a bit of an overview of cultural and biological type practices. But what I wanted to do is announce this. So I'm a part of a federal effort looking at fungicide resistance. It's a pretty big cornerstone of my research program on campus. And but I'm not going to talk about it, fungicide resistance at all today. Maybe I'll mention it, but I wanted to announce this workshop. It's a collaborative between Minnesota, Wisconsin, and Michigan. And we're going to have an announcement from Michigan State Extension Service coming soon. But we have a website called Frame Networks, and it's on March 2nd, 3rd, and 4th. It's there's, I think, 25 or 30 slots per day. And we're trying to get growers from some of the different states. And we're going to pick 2 diseases and sort of compare and contrast the best way to make a spray program to control for them. I believe we're thinking powdery and downy mildew this time, but I don't know if we've fully made that. We have a workshop that we plan on moving around the country. I've been a part of building it. So I just wanted to announce those 3 dates coming up. So this talk is really focused on some of the major diseases that we'll see in grapes. And I kind of do an overview of just the cultural practices at first, and then a bit of a review of what fungicides are, and then what biological fungicides are available in vineyards. And then at the very end, I have a little trial that I was going to show. So nothing, I've shown it a little bit before in different ways, but I just want to highlight some of the new products that are out there. So Phomopsis, and I feel like this is a huger issue in the southern part of the state, but growers do experience it. It's usually considered an early season disease, and cultural practices include removing diseased canes. So for example, within our research block, we'll purposefully leave a bunch of old, dead, nasty canes just so we have a fair amount of Phomopsis inoculum. So removing those diseased canes can be quite helpful. Anything that improves air circulation will also help. And really between 1 to 10 inches of shoot growth is kind of the critical time if you see those pitting of canes and things like that. But generally speaking, and I said this is all cultural, protectant fungicides do work fairly well at controlling this disease if they're available to you. So black rot is the next disease, and that's sort of similar in some ways. Again, it's old plant material in the form of mummies that are on the floor from the previous season. So anything that removes mummies, good weed management can be helpful against black rot. Again, anything that reduces or increases airflow in the canopy can be quite helpful. And it's sometimes at a fruit stage kind of hard to tell the difference between black rot and phomopsis. Generally, black rot will happen first. It'll kind of engulf the berry, sort of. The lesions on the leaves look quite different, though. Typically, black rot lesions are larger than phomopsis. But in many ways, these guys are similar because they're both early season diseases. Powdery mildew, cultural practices, there's not a lot, but anything that improves airflow, increases exposure, reduces wetness can be helpful. So anything to that effect. There are cultivars, and it's a long-term game plan here, but trying to get cultivars that are resistant to powdery mildew, or at least somewhat tolerant. The big issue with powdery that most are concerned about is that if you let it go for a long time and you get a lot of fruit infections, it can cause the fruit to russet, get scaly, crack, and that can lead to other problems like bunch rots later in the season. Yeah. Downy mildew can be considered one of the more critical diseases in Michigan, mostly because of our harsh winters. So it's a major disease. It's caused by a completely different organism. So downy mildew is not it's not a fungus at all, even though we call the things that control it fungicides. Not primarily it's not a fungus. It's actually related to diatoms or algae. It's more related to plants than fungi, but it kind of behaves like a fungus. So anything that decreases wetness, so these are considered water molds often, can be helpful. You're kind of concerned about two parts when you think about downy mildew. It's the fruit infection followed by the foliar. And the fruit infection is, I'll show a slide of when that mostly happens. But in Michigan, the foliar you're concerned about probably all the way until the end of the season, or at least until like mid-September or so. Because if you have the premature defoliation, starch won't necessarily accumulate as well in the vines, and then if they're in a place that gets really harsh winters, the vines could die. So that's sort of the critical aspects of it. The defoliation can be pretty dramatic. So this photo, even though it looks like late fall that I took this photo, this is really bad downy mildew infection, probably as early as, I think I took this like September 1st, and that's was way too early for it to look like this. They hadn't had any frost yet or anything. So luckily this was in an area where these vines would probably have been okay just because they were really close to the lake, but in general can be quite devastating on the winter hardiness of vines. So downy mildew is just a few things about downy before I go further. Rainfall really drives these epidemics, and that's what makes downy mildew epidemics worse, essentially. So if you have a really rainy, heavy dews all summer long, you can really get more and more downy mildew that can accumulate. But for the fruit infection, growers out west where they have really mild climates where they occasionally get downy really only care about the fruit infection. And that you can see is pretty much right around bloom. So about 12 days pre-bloom, bloom, and then about 7 days post-bloom, depending on what variety we're talking here. It's sort of this critical period where you can get fruit infection. But later in the season, you won't see fruit infection anymore. You'll just see the foliar. So that's kind of how downy mildew works. So kind of moving on to another disease, botrytis bunch rot. Again, sounds like saying the same thing over and over, but essentially anything that reduces water can help against botrytis. If things get overwatered or they are getting overhead irrigation in some way, it can really drive botrytis infections. So good water management and fertilizer is important. Preventing any kind of berry damage, whether it's mechanical with a tractor or bird packs or insects, can help with botrytis. And there are some varieties that ripen at different times. So paying attention to when you use water relative to when the fruit ripens. We have a cultivar called Aurora we use a lot for botrytis trials, and it ripens really early, like mid-August in southern Michigan. But the fruit will split because it's still raining quite a bit, so it just gets a lot of botrytis. Anything that manages the canopy to reduce that water is also important. We do have a new fact sheet from MSU that might be helpful to kind of peruse over if you're not super comfortable with botrytis. It's part of a series that we're launching at MSU for different insects, diseases, and probably add a few more things to it. So we've got about 4 of them right now. And I didn't really have time to go over crown gall, but crown gall is one of them. We have leaf roll viruses are another, but we have this one for botrytis. So sour rot's kind of an interest right now in our program because it also seems to be a pretty important bunch rot. If you're growing a cultivar like Vignoles or Pinot Noir, that's got a really tight cluster, you can get a fair bit of sour rot if the conditions are right. Fruit flies, yellow jackets can be responsible for movement in the vineyard. It really develops when you have warmer temperatures. So sometimes it can be a little more severe in the south part of the state. And it really happens when you get above 15 Brix. It's been associated with a lot of different bacteria and yeasts on fruit. It's sort of an interesting research question, but trying to control it's a different objective here. But essentially, there's a lot of different organisms that go after this, and it gets that sort of wet, slippery, and then this sort of fermented smell in the fruit. We did do a trial, and this has some conventional products in it, but it has one that is a organic one that's a softer product that I wanted to highlight here. So this is a trial of organic and conventional options, and this builds on some work that was done at MSU and at Cornell, looking at what insecticide / sterilant fungicide should be applied near harvest that you could essentially get good control of sour rot. So this is in a site that has extremely high levels of sour rot. So it's Vignoles we've been working with for a while. And fracture is a lupulin extract as a compound. And that's that one or oxidate seem to be the better fungicide components to that. And the research that's been done at Cornell and other places sort of showed that an insecticide can be really important during that. So we were trying to figure out if we could do different fungicide / insecticide options to get good control. So what I highlighted in the box here is the fracture by itself. So that's no insecticide being applied with it, and it's a lupulin extract, and we got quite a bit of control. It's not perfect control, not what you would see with fracture and Mustang Max or anything, but we did get probably 60% control. I didn't do the math here, but we had overall severity was about 55%, and in this one it was more like 20%. So a fair bit of control. So anyway, I just highlighting that because we're just focused on more softer chemistries here, but I think that's an interesting result. Also, before I skip into the fungicides, I just want to say that scouting can be really critical, and figuring out what you have in your vineyard is really important. Earlier this last year now, we turned the pocket guide that MSU has produced into a mobile app. So it's freely downloaded. There's a few MSU articles talking about it. So feel free to peruse and check that out. I think it's worthwhile to take a peek at. And it's nice because it fits into your phone. It's really just a clickable PDF, and you can just click on these different links and it ports you all around the scouting guide, and that can be pretty useful. So that's the pocket guide. We also, I think at MSU, are trying to do more grape scouting reports. We're trying our best to make these more statewide. Mike Rinke, who's our new educator, did a really, I think, great job leading us as a team, probably herding cats in some ways. But we've tried to get this a bit more of a statewide effort. And we have the entire grape team is pretty much on this. So we put reports out every week. And this is one was our last issue, I believe, at the end of September, talking about what we saw were issues, goes through all the diseases, all the insects, some of the horticultural tips we're trying to add more to. So I think it's quite interesting as a scouting report. So we're worthwhile to take a look at. And I believe we're hoping to keep that going this year. It's on our MSU grapes.msu.edu page. So what I wanted to do is switch and just talk about, so I guess I'm somewhat known for talking about fungicides and fungicide resistance, and that's sort of the big part of our program. So I kind of wanted to back up and talk about what a fungicide is. So, fungicide is a biocidal chemical, could be biological, and it basically kills fungi and their spores. So it's a chemical barrier on the host plant, and it can eradicate an existing infection sometimes. So generally speaking, pesticides aren't very good at curing any diseased plants, and that's definitely true for some of the biologically based ones. They're going to be not so curative at all. But so this is sort of what a fungicide is and why we use them, why they're a good tool. And so how do they help? I kind of want to start off, if you've probably never or are bored by the disease triangle, but I just want to show you that, disease only happens under certain circumstances. It's actually not a common phenomenon. Microbes are all over every surface on every part of the planet, but they don't cause disease very often. It is this sort of trifecta of things that have to occur where you'll get disease. And so if all the stars align, I guess, you can think of it that way, you'll occasionally get disease that will develop. And that can negatively affect your plants. So just to point out, anything that we can do that would mess up the way this environment is or the pathogen directly will destroy the disease. So that's really, you're either making the environment inhospitable or you're disrupting basic cellular functions of the pathogen. Either way, it's shutting this disease down. So fungicides have been around, and grapes have played a huge role in the development of fungicides. So this is just for fun, I figured I'd show this. This is a historic look at some of the different fungicides and how they've been developed. Started off with brine, people have used arsenic, and then finally copper sulfate, sulfur, lime sulfur, and so forth got developed over time. And grapes, probably around 1824 going forward, played a big role in developing some of these products. Bordeaux mixture is pretty famous, and it was actually used to deter pilferers originally, and then basically they sprayed it and it caused significantly less downy and powdery mildew. So this became the common mixture that was used in France. So mildews has had a huge impact on the industry. There was a period where they lost probably 80% of the French crop due to powdery mildew. Downy mildew became a problem in 1882 throughout France. And then there were several years, which they call the mildew years, which don't sound fun at all, where this Bordeaux mixture, they were trying to figure out how to use it and how to use the timing and get growers to adopt it. But there were a couple decades there where they lost significant crop due to both these two diseases. But since then, this particular mixture has played a big role in protecting Europe from both the mildews. So anyway, backing up, I want to talk a little bit about fungicides and how they are. So there are contact fungicides. I know this has got a lot of text in it. There's contact fungicides, and then there's systemic fungicides. And contacts basically are there. Some examples might be mancozeb, coppers, biofungicides, Captan, and all these products are there. They'll inhibit the pathogen, but then they will go away. There are systemic fungicides, and these, a lot of the newer ones do sort of get into the plant and stay there and move around. So generally speaking, and I told you this is all about soft products generally don't do a great job of being systemic. There are some examples where they would turn on defenses. So newer ones like Lifeguard, I put a giant jug of Regalia here, potassium phosphites, Profit, and some of the biofungicides may do this, but they would only do it because they would trigger SAR. The chemicals and metabolites aren't very systemic, but if they trigger SAR, this process can kind of cascade through the plant and cause resistance to form. So generally speaking, a lot of the biofungicides, though, are contacts. When you think about fungicides, there's different ways to look at it. They can be preventative, they can be curative, and they can be an eradicant. So preventative means before infection, curative means after or back action. Bio or soft products are generally not very good at this at all. And then there are some examples of things being eradicants. So potassium phosphite or Profit, which is a softer product, can do a pretty good job of eradicating and burning it out, as some people might say. And generally speaking, but most of the time your softer products, and this is true of even the more site-specific ones, are better when they're applied preventatively before the infection happens. And this is just an example of when in the stage. So preventative is used here during this infection. Mycelial growth will be a little bit here. This you'd need a curative product if one existed. And then this is more of an eradicant time. So this is like going to dry your leaves out and basically, you know, burn it out, as they might say. So that's just sort of how you might classify fungicides. One more definition of it. So when you think about it, there are different types of activities. So you have single site fungicides, multi-site fungicides, and then they have, and this basically means it affects one specific thing. Most biofungicides are not single site fungicides. Most softer products are not. Most of the organic and biofungicides would be considered multi-site fungicides. Things like copper, and many of the biologicals have both a pesticide component and an SAR component to that multi-site thing. Profit, which I keep using as an example, has both a fungicide component where it's killing the pathogen in some way, but it's also triggering SAR in other ways. So that would be considered multi-site. So that's one way to classify activity. And the other way is narrow versus broad. And narrow, again, works probably better for some of your single site fungicides, like I'm thinking Elevate for botrytis. That's a very narrow spectrum. It really only works for botrytis. The broad spectrum, meaning it works on several things. So copper works on a lot of products where, you know, and then that would be meaning it's working on many different individuals. And I stuck this big picture of mushrooms here because a broad spectrum fungicide would kill all these mushrooms, but a narrow spectrum would only kill the one that has sunglasses or something. So just as a little bit of a fun example. So we use this product list called OMRI that historically just, I should start off, organic fungicides historically actually just meant fungicides that had carbon in them. So you have to kind of be careful if you want to look into that history stuff. But now it's regulated and it's set up by this OMRI list and what's considered OMRI. And it basically means food that's grown without artificial chemicals, hormones, antibiotics, or genetically modified organisms. And that's sort of the general way to think about what organic food is. Anyway, most of these products are better as preventative, and they need to all, usually under high disease scenarios, need to be applied sort of regularly. And that's sort of the double-edged sword with them in that sense. You have to apply them quite often. Generally, and I don't have time to go into a lot of detail on the specific groups, but there are several ones that this list that OMRI puts together is a little bit confusing, but you basically have biopesticides, botanical pesticides, fungicides, microbial pesticides, and I would group that in sort of the same thing, but the list breaks that up. They're not all the same, but the list is a little bit hard to interpret. But essentially, these are biologically based pesticides in some way. There's calcium polysulfides, copper sulfate, coppers, oils can be used. Particularly oils tend to be quite good on mildews. Lime sulfur, hydrogen peroxide, also as an organic. Some different oils like JMS stylet oil, and then potassium bicarb or caligreen is also a kind of one. So this is sort of some of the main tools that you'd have that would be listed in OMRI for grapes. So in general, and this is just efficacy, soft and organic products can be effective, and it's possible to grow grapes in Michigan organically. That said, generally speaking, they're not as strong as their conventional site-specific fungicide counterparts. In grapes, the majority are better on foliar pathogens. So like oils actually tend to be quite good on downy and powdery if they're used correctly. You have to be careful of phytotoxicity. Probably the most difficult diseases are going to be the bunch rots. So botrytis, sour rot are pretty hard to control. And that's probably even true of with some of the site-specific fungicides because there's so much fungicide resistance. But some of the bunch rots are going to be the more difficult diseases to handle. Generally, they need to be applied regularly, so not as long of intervals maybe as the conventional counterparts. And usually they need to be applied before. So some of the one products, and I know I'm running short on time, but some of the other products that trigger SAR need to be there about 5 to 7 days before infection starts. Because of the desire to grow organically though, It doesn't mean that you have to use all or only soft and organic products. A lot of these products actually have a good place in conventional programs, and that's, I think, probably the way to ease into some of these products, because we have so much fungicide resistance, and we have these products that do work. I think that they should really be thought of as a different mode of action that's available to growers. So that's just something to think about that could be incorporated into a more conventional program. There are a couple new products that we've been working with for a few years. Stargus, which is made by Marone, Lifeguard is made by Certus, and Howler is made by AgBiome. And they all are bacillus-based products. Lifeguard is more of an SAR product. Stargus is more of a biofungicide, and so is Howler's more of a biofungicide. And I don't have a bunch of data because I knew I was going to run short on time. But this is a downy mildew trial we did this year that is an untreated, and this was in Niagara. But we get quite a bit of downy mildew pressure in our Niagara groups. And this is just to show we had a lot of downy mildew pressure, almost defoliated the plants. And with Lifeguard applied, probably about 6 treatments of it, we had very little downy mildew, and also for other diseases too. We used OSO, which I'm not going to talk about that right now, but we used Lifeguard in a program and Howler in a program, and we had an industry standard. So essentially, we had a pretty good control when we used it alone, but then when we used it in a program, it worked better, and so it did Howler as well. And sorry that Stargus isn't in here, but we've had some good results with that as well over time. So when you're thinking about doing a pesticide application to control diseases, whether it's organic fungicides or conventional, just try to think about these few things. Timing the application, make sure the pathogen's active. Is it going to rain? Just think about the biology of the situation. Coverage issues can be important, especially when you're working with products that might not be as strong, contacts in particular, because they're not moving around the plant very well. So they really have to be there where the pathogen is going to land. So things like, the amount of gallons per acre, how fast are you driving, calibrating the nozzles, working on droplet sizes. You don't want it to be too small. And then like water quality, there's a lot of fungicides that are pretty sensitive to pH. So I think it's better to think about these usually preventatively, and then so it's nice to think about the biology of the situation, but also that physics of actually making an application, so hitting the target. So I've had a lot of support, and I realized this was not a very heavy research talk. It was more of a going through some organic products. My group's pretty focused on a lot of research questions. We're doing a spore trapping project that the Wine Collaborative has been supportive of. We're also doing fungicide resistance, but I just wanted a chance today to talk about organic pesticides. So with that, I'll take some questions. I don't know, I was on a Star Trek theme when I was putting this together, so well, there's grapes here. So anyway, I'll take a few questions though. So.
Thank you, Tim. It was great presentation. I'm really interested in this topic, biological control. Here, growers, they are very dependent on the clinical control of pests. So, and I believe we can run a good educational project to enhance the knowledge of the grower about the biological control. Thank you so much. Annette, could you please read the question for?
Yes.
Tim? Yeah, thank you.
Thank you, Tim. That was a great presentation. And a kudos in the Q&A box. Slide art is a 10 out of 10. We are a little short on time, so I'm going to just pick a few questions that are specific to your talk today so you can expand on that. And then we may have to answer some of these questions by email follow-up.
Okay, I'll try to answer as quick as I can, or might be fast.
That's great, thank you. Someone asked, lupulin is very gummy and aromatic. Any concerns about residual on the fruit going to the wineries?
I don't think those trials have been done. Had really good results for sour rot control though. But yeah, I don't think I've done that kind of work. So.
Okay. There was another question. What was the other fungicide that was like fracture that you mentioned? Fracture is very hard to find.
Oxidate 2.0, and that's a group. Cornell was focused a little more on that. We kind of had two competing things going, not competing, but different sets of trials going on. And Cornell did Oxidate 2.0. I believe that's what they would recommend. We did more work with Fraction, and I think that's FMC's product. Oxidate's Biosafe Systems, but we both have found good efficacy with both of them. So.
Okay, thank you. Do you do the sour rot trial with just Mustang Max and no fungicide?
We've done that as well. I think I took it out of the slide, but we do it. And in general, and this is a bit summarizing some of the Cornell work, but that insecticide was quite important. And that's why we're not getting that perfect control, because the site that we're at has quite a bit of insects. It's right up against a tree line, and there's a lot of insect pressure. So I would imagine we'd get similar results. That said, the fracture alone did pretty well. But
okay, thank you. Do you have grape phenology and related IPM developed?
Do I have grape phenology and related
insect pest management developed?
Somewhat in our fruit management guide, I suppose, is the way to couch that question. But it's also in the pocket guide. Down the road, if there's points that people think that need to be edited, though, I'd be editing things. But we do try to edit that in the fruit management guide. And then in the scouting reports, it's kind of a more of an active way for me to communicate with you guys.
Okay. Have there been studies on how Bordeaux mix affects soil? Is it mycorrhizae?
Mycorrhizae.
Mycorrhizae, thank you.
I think as soil gets, its pH gets altered, and there could be toxicity levels once. So I believe there has been, but I don't have them fresh in my mind. I think, decades long of applying that similar mixture of lime and sulfur may have some deleterious effects on soil, but I don't have those studies right on the top of my head. I know they use it for several decades, or they still use it, but yeah.
Okay, thank you for that.
Mm-hmm.
I think that's the last question we're going to take, because we need to get on to our next talk. But for anyone who's hanging on, if you want to continue type questions in the Q&A box, we can definitely have Dr. Miles answer those and email them out to our participants. Thank you.
Thanks, everybody, for the opportunity.
Thank you so much. I appreciate your time. Thank you.
Mm-hmm.
So let me share the, this is RUP code. Just make sure you just jot down this code. And also we have a survey. It takes 30 minutes. I'm kidding. It takes only 10 seconds. And your answer really help us to evaluate our job. So thank you so much if you answer those questions. Only I think it's a 3-4 question. Thank you so much.